Okay, so good afternoon, everyone. My name is Ayat Shoaib. I'm a second year medical student, and I'll be teaching you blood gas transport today. And I think this is your P25. So if you have any questions, you can go ahead and email me or text me on my number. Okay, so what we're going to be covering today is basically the starting experiment the doctor did, the physically dissolved oxygen and the chemically bound oxygen. Then we're gonna see the factors that affect the oxygen curve. These are some of the factors, and then the forms of carbon dioxide and the factors affecting its curves. Okay? Okay, let's start off with what you guys have already done before. You guys have taken the gas transport in and out of the lung through ventilation. Then you guys have known how gas diffuses through the alveolar capillary membrane. Right? Now what we're gonna do is you see this blood part, we're gonna see how carbon dioxide and oxygen, they're transported with the blood to different organs. Okay, and they come back. Okay. So this is what the doctor started with. You don't need to know this process, but he took whole blood and he put it in a centrifugation machine. What this does is basically goes round, round, round. I think you took it in the mole lab, I'm not sure yet. But okay, it separates the parts of RBCs and you get plasma, you get white cells, and you get RBCs. Okay, now what the doctor did is he removed the RBCs or the erythrocytes and he took just the plasma. Okay, and I want you to remember this one thing that hemoglobin is needed for gas particles to bind. It's like a goblin, right? It wants everything to itself. It wants to bind to something. So it's needed for gas particles to bind. Otherwise, they're just unbound and free, okay? They escape the goblin. And hemoglobin is present in RBCs or erythrocytes. And what did we do? We removed the RBCs. So we don't have hemoglobin. So all of our oxygen is going to be free, okay? So this was the picture he used in his lecture. This is our plasma, free of erythrocytes, no hemoglobin. Okay, so all of our oxygen, let's assume the gas phase, everything here is oxygen. All of our oxygen is free. Okay, now as soon as, one second, okay, this is our oxygen. Okay, as soon as this oxygen touches this surface, it's gonna be pulled in. Okay, it's soluble, so it's gonna be pulled in. It's like this clown. Like, sorry. It's like this clown. It's just gonna pull everything in. Okay? So as the oxygen molecules, they move in, the partial pressure increases. Because as I said, there is no hemoglobin here, right? So oxygen molecules, all of them are free, right? They're moving anywhere they want to. They're exerting their own pressure. So here you have oxygen particles. They keep moving in. More particles. It just increases the overall partial pressure. So here I said more PO2 in the gas, more goes in, more PO2 in the fluid, more pressure in the fluid. Okay. Uh, at some point, this movement is going to reach an equilibrium or steady state where the PO2 over here is going to be equal to the PO2 in the fluid phase. Okay. There's this thing, I think we're all done. Example, you have two balloons, right? Oh, my bad. Okay, you have two balloons, right? Assume this has 70 particles and this has 30 particles. Okay, so you know what diffusion is, right? Diffusion is the moment where there's high concentration and low concentration. It's going to move from the higher to the lower one. So it's going to keep moving from 70 to 30, okay? It's going to keep moving until you have like both of them are equal, which is going to be 50 and 50, okay? Now the thing is, the air around us, it just keeps moving, okay? So the oxygen molecules, they can keep moving back and forth. Like we're not freezing them. Like we're not saying stop. They keep moving back and forth, but the net movement does not change. So there is oxygen molecules moving here and they're moving here, they're moving here. But overall, it's still 50 and 50, okay? It's not that important, but just understand. Okay, we can calculate both partial pressure and concentration of oxygen in the fluid phase. So as I said, more PO2 here, more of it is pulled in because solubility. If it's pulled in, there is more pressure, and also there is more concentration because you're taking more oxygen from there and putting in the plasma, right? So you have more concentration overall. Okay. Another thing is that physically dissolved oxygen. What is physical, right? You can see something. You can see a bottle, you can see another person, you can physically see them. So this oxygen, you can physically see it. You can't really see it, but you can see it there Like it's not bound to anything. It's free. It's on its own, right? It's its own person. So it's non-bound oxygen or physically dissolved oxygen. Again, as I said, because as the pressure is going to increase, as the pressure is going to increase, the concentration increases, right? As I said in this experiment, if the pressure increases, that just means overall particles increased because the pressure increased. That just means concentration increased. Again, here concentration is directly proportional to solubility coefficient because if it's more soluble, more particles are going to enter and the concentration is eventually going to increase again. Okay. Do you guys have any questions? I'm gonna assume no questions. I can't really see the chat, by the way. Okay. Okay, so oxygen is of two forms. One is physically dissolved. This looks like a Michelangelo painting, but anyways, it's physically dissolved, right? It's its own person. It does not bind to. There is basically no hemoglobin in plasma. But anyways, there is in the blood too. By the way, there is physically dissolved oxygen. It's not just in the plasma. Even in the blood, like with RBCs, there is physically dissolved oxygen, and it's not going to be bound to hemoglobin. So it's unbound and free, okay? And there is chemically bound, which is bound to hemoglobin. So chemically bound is like, you know how in chemistry you have bonds between molecules, so they form a bonds and they're like inseparable and stuff. So yeah. Okay, so partial pressure. This is another thing you really, really need to know so you don't confuse these two terms. Partial pressure is only produced by free molecules, okay? As I said, they're their own people, they exert their own pressure, they're not bound to anything. So partial pressure is only produced by free molecules, okay? However, concentration is going to be the total number of free molecules, which means unbound, or bound molecules. So it's both chemical and physical, okay? Because, okay, for example, you have, these are like free molecules, okay? They have their own partial pressure, they exert their own partial pressure. And then over here, you have, this is hemoglobin, and you have, this is oxygen on it, and this is free molecules. Okay, here the concentration, you're gonna count both. You're gonna count the free molecules and you're gonna count the bound molecules, okay? So it's gonna be both. Remember this. Okay. I'm gonna start off with this. So equilibrium between physically dissolved and chemically bound gas. In this one, we just have physically dissolved. So you can see over here, there's just free molecules, right? Physically dissolved, non-bound, free molecules. There's free molecules and there is 8 of them. And even here, there's 8 of them. So the partial pressure of gas A is equivalent to the partial pressure of gas B. Why? Because the free molecules are equal. What about the concentration? The concentration is also 8 and 8. So that is also equal. Okay? Partial pressure is equal. Now when we move ahead, over here, I'm going to add hemoglobin. So what my oxygen is going to go do is it's going to go and bind to the hemoglobin, okay? So because it's bound to the hemoglobin now, it does not exert its own pressure, okay? It's kind of jailed. It has no say in anything, okay? So it's gone. So our partial pressure again is free molecules. Here you have 8 molecules again. Here you only have 2 molecules, okay? So here, partial pressure of A is greater than the partial pressure of B, okay? Partial pressure of A is greater than that of B. But over here, the concentration, I don't know if you guys can guess the concentration. Concentration is both bound and unbound. So here you have 8 free, here you have 2 free, and 6 bound, okay? Which is equivalent to 8. So concentration is still the same here because, again, concentration is both bound and unbound, okay? We're gonna use the principle of diffusion here again. So a driving force, you need a driving force for O2 to move. So here in A, you have a higher partial pressure. In B, you have a lower partial pressure. So eventually the particles are gonna start moving from A to B to equalize the pressure. And then you're gonna get this. Okay, so all of my free molecules, they moved from A and they went ahead and went to chamber B. Okay, so when the oxygen increases here, they kept binding to hemoglobin, and eventually this is what we're left with. Over here, you have 2 free molecules. Here again, you have two free molecules. So partial pressure, it goes back to being equal, okay? So there is an equilibrium with partial pressure again. That's what this means. But the concentration here was same. The concentration here was same. It was 8, 8. It was 8, 8. But here, the concentration, here you only have 2. Here you have like 6, 12, 14, okay? That's a big number. So the concentration in B is way more than the concentration in A, okay? Then you have more free O2 is more bound to HB. Okay, so this is what we're going to take later. So if you have more O2, like you have a lot of O2, so it can easily bind to hemoglobin. If you have less O2, hemoglobin releases it. We're gonna take it later in a bit. Okay. Here, physically dissolved O2 is less, as you can see. Even though it's quantitatively less, it's only 2, but functionally it's important. Another thing you need to know is only three forms of O2, it can go to different compartments. So if I have bound hemoglobin, if I have oxygen bound to hemoglobin next to a tissue that really, really needs O2, I cannot give it to it until it releases the hemoglobin, right? I need it to be free so that I can go and give it to my tissue. If it's bound to hemoglobin, I cannot give it to the tissue, okay? And eventually the tissue, it's gonna be like it's gonna go hypoxia and all of that, and eventually it's gonna die because there's a lack of oxygen. Um, okay. Okay. So there again, there's two forms of O2. We went over this. Okay. This curve, it's kind of confusing for a lot of people because it has two curves in it. It has the, if you can see over here, let me change my color. Okay. If you see over here, it has the red one. It's the physically dissolved curve that we just took. And this one is the chemically dissolved one, chemically bound one, okay? As I said in the physically dissolved one, as you increase the partial pressure, the concentration increases, okay? Also, when you increase the concentration, like more oxygen, more of it binds to hemoglobin, okay? So at maximum volume for chemically bound O2, all binding sites are occupied. This means that if you keep increasing the O2, like if you keep increasing the concentration, at some point all of the binding sites are going to be occupied, right? Like at this point, all of the binding sites are occupied. And due to this, the lines from both graphs run parallel to each other after a certain limit. Like you can see this line and this line are basically parallel, right? Another thing is that if you keep increasing the pressure, you can still get some of the oxygen to go and bind to hemoglobin, but it requires pressure, okay? You can't just, it's not as easy as it was like over here, okay? You still need like a lot of pressure for it to go and bind. So it gradually increases, like you can see it's gradually increasing with the physically dissolved one, but it's not as steep as this one, okay? Um, so this is our total oxygen, obviously, because we have the physically dissolved oxygen here, and we have the chemically dissolved oxygen in the orange side. So how would you calculate the chemical one? You would take the total, which is the whole thing, the green part, and you would subtract the slope below. You would subtract it from the whole thing, which would leave you with the orange part, which is our chemical one. That's all in this slide. Okay. Another term that the doctor talked about was O2 capacity. And O2 capacity is basically related to oxygen that is bound to hemoglobin. Okay. So O2 capacity is basically when you have so much O2, you completely saturate all of the hemoglobin, right? It's the maximum volume of O2 that is bound to hemoglobin. And after this, you can't bind with hemoglobin, like usually you cannot bind with hemoglobin anymore because you occupied all of the binding sites, so you can't really do anything and you can't bind with it. So your all your capacity is basically filled by oxygen, okay? There's nothing you can do. So it does not consist of physically dissolved. Obviously, so it does not consist of physically dissolved because physically dissolved is not really bound to hemoglobin, and we're just talking about the oxygen that is bound to hemoglobin. So it does not consist of physically dissolved. Okay. Also, it's not just any point in the orange part. You can't say, oh, this is the maximum value because you can see that it's still increasing, right? And we said that auto capacity is the point where you basically cannot increase anymore, like over here. It's taking a lot of pressure to increase it. So that means like all of the sites are basically bound. Okay. So when there is more oxygen, if you have more oxygen, it dissolves in the blood and the hemoglobin goes and binds there, right? More oxygen, it's easier to go and get it. Okay? So the amount of both dissolved oxygen and the oxygen on hemoglobin goes up together. Okay? Like as you can see here. If I keep increasing the concentration, it's just gonna keep going. Like they keep going together, right? And sorry, these lines go up parallel together because I keep increasing the hemoglobin. Sorry, I keep increasing the oxygen, so the hemoglobin continues binding to it, and there is overall free oxygen too. After if there are no hemoglobin sites left, the oxygen is just free. It just moves everywhere. Okay. So this term, affinity is how strong you attach to something. It's like this really, really strong friendship, okay? So for example, you have our whole lecture is around hemoglobin and oxygen and gas, okay? So when you have hemoglobin, it has a very high affinity for oxygen. It easily picks it up and holds on to it. So it kind of like, it has like a really, really strong friendship with it. It just does not let it go unless like there's a conformational change or anything. Okay, we're gonna talk about no. Okay. Okay, so first in this curve, you can see that there is saturation here mentioned. Okay, if you notice in our previous curve, we talked about concentration against partial pressure. Here we have saturation. Okay, saturation is basically like you have a hemoglobin molecule and it has four binding sites, okay? It has one binding site here, one binding site here, one here, and one here. If all of them are occupied, we say that this molecule is 100% saturated, okay? If I only occupy like these two, I'm going to say that it's 50% saturated, okay? Another thing is, okay, so I have like, let's say I have four hemoglobin molecules, and I occupied one, two, three, four. I occupied only two of them, so there's gonna be 50% saturation. Okay, so that's how it goes. Even like this, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. There's 16 available sites to bind, and I only went here and here, and I went here and here, and I went here and here, here and here. I occupied only 8, so 8 by 16 is 1 by 2, which is half, right? Which means again, I occupied only 50%, so only 50% is saturated, okay? So first we're gonna go to a pressure of 27 mmHg. So this is a normal pressure in capillaries. Okay. Normally in capillaries, only 50% of hemoglobin is saturated. Okay. And then if we increase the pressure, as you can see, we're increasing the pressure, the saturation just keeps increasing, right? And okay, let me talk about this first. So if I need a high P50, okay, I need a high pressure to attain 50% saturation. Okay, I need a high pressure to attain so I'm going to increase the pressure. First of all, high pressure to attain this one. It's going to go here. So it moves to the right. Okay, what does this mean? It means I have a low affinity for O2 because I have a low bond with O2. It's like, oh, I don't want to, you have a low affinity for physio. I don't want to do physio. And then you just leave it for the last. And now you're like, oh no, like you have a lot of pressure on you, right? You need a lot of pressure for low affinity. Okay. And more pressure is needed to occupy. This is basically more pressure is needed to occupy 50%. And the curve shifts to the right. Okay. You need low pressure for 50% when you have high affinity. So you have a really strong bond. Like, I like this so much, like I want it right now. You don't need a lot of pressure to do that. You can just do that as you like, right? So for this, less pressure is needed to occupy 50% of the binding sites. It's at zero. It's okay. You go left and you go 50%. Okay, because you don't need that much pressure because you have a very high affinity for oxygen. You like oxygen very much, so you don't need to be pressurized into taking it, okay? You're gonna willingly take it. Okay, so here 27. Okay. Over here a pressure of 100 mmHg. I think you studied this yesterday, I'm not sure. It's at the end of capillaries. If we ignore the right left shunt, it's also the arterial O2 partial pressure. Okay. Basically at 100 mmHg, all the binding sites are basically occupied. So we say there is a full saturation. Like you can see here, again, it's a flat curve. So there is no more available sites for hemoglobin to bind. So it's a full saturation, okay? That's basically what it means. There's 98% saturation. This isn't point, but when you change it to percentage, it goes 50%, okay? Don't get confused with that. Okay, that's math. Okay, pressure of 40. Okay, this pressure is present in the mixed venous blood, which enters the pulmonary capillary. Okay, so this blood, it has a 75% saturation. Okay, it already has oxygen. It has 75% of it already has oxygen, and only 25% has to be saturated. So this is kind of crazy, right? Because why does it even come to the lung if 75% of it is already saturated? Like it doesn't need it, right? The point here is you have let me go here. Okay, all of the parts of your body, they need different amounts of blood depending upon what they're doing. So for example, your heart is always pumping the blood, so it uses like 70% of the blood that's being pumped to it, and you only have 30% left, 30% oxygen left. Okay, in the kidney only uses 10% of the oxygen and it gives back 90%. So different parts of your body, like the brain uses more and it leaves the RBCs with less oxygen saturation. All of it drains eventually into the right ventricle, and then the average is 75%. It's not like that the heart uses 25% and gives you 75%, the kidney uses 25, 75. No, all of it averages to 75%, and it drains into the right ventricle, and from there it goes to the lungs to eventually be saturated, the 25% saturation, and then it's fully saturated, and then again it goes back to the tissues, and it's the whole respiration cycle. Okay, so saturation can also decrease during exercise. Okay, there is a curve for temperature that we're going to study about later. But just know that hemoglobin is like a reservoir, okay? So it always has oxygen bound to it, okay? It's like best friends with oxygen. So it always has oxygen bound to it. And whenever somebody needs help, it gives oxygen away. Like, oh, here you go, okay? If your tissue, like during exercise, your muscles require oxygen, right? So during exercise, more oxygen is used up. Why? Because the blood vessels near it, they realize, oh, I need to give it more oxygen, so I should release more oxygen. Okay? So it starts giving it more oxygen. It starts releasing more oxygen, and more oxygen is basically given out to the muscles. In the flat part. Mm, okay, so in the flat part of the curve, the amount of oxygen bound to hemoglobin stays nearly constant, even if PO2 changes. Okay, we can see if the PO2 is changing in this part, it's still flat, right? So this helps maintain a steady supply of oxygen to the tissues, and this helps us protect against hypoxia and ensures that the body can function well in different conditions. Um, because if this part is kind of damaged, then you don't really have 100% saturation, especially in the lungs, and then you don't really get enough oxygen to other parts of your body, and that can cause like low oxygen levels. Mm, mm, mm. Okay, in the steep part of the curve, small change in PO2 is huge change in saturation. Because as you can see, over here, it's here, right? At this partial pressure, the saturation is this much. But if I increase it this to 40, it increases by this much. So unlike here, which is flat and there is not really much change, here there is a big change. Okay, this is where blood flows in tissue capillaries. It's like, okay, so if you have a blood vessel and you have a tissue. The tissue has less oxygen and the blood vessel has more oxygen. So again, if you use diffusion, the oxygen is going to move from the blood vessel to the tissue, okay? From a area higher to a area lower because your tissue needs more oxygen, right? Okay. Um, go ahead. Okay, I want you guys to answer this if you try and answer this if you can. So hemoglobin. For example, let's say I had 4 molecules of hemoglobin. Okay, and I reduce this to only 2. Mm, can you tell me what happens to the this curve with concentration? Okay, this one has saturation, this one has concentration. Can you guys guess? Okay, so basically, the concentration here, in the concentration curve, it's going to decrease. The concentration curve is going to decrease. Why does this decrease? Okay, okay. It decreases because you had four molecules of HP, which means you had mm, you had one, two, three, four. You had 16 molecules of oxygen here. Okay, so you had 16 molecules of oxygen here, but you reduced them. So you have only eight now. So your concentration is going to decrease, right? Because I said concentration. What was concentration? Concentration was both bound and unbound. Here, when we talk about just bound, you reduced it, right? You reduced it from 16 to 8. So your concentration eventually decreased. When we come to saturation, saturation is going to stay the same. Why does this happen? Because again, you had four molecules here. Let's assume they're completely saturated. Saturation is going to remain the same. This is why. Okay, you had four molecules before. Okay, you decreased half of them. In these two molecules, the oxygen, it occupies all of the binding sites. Okay, so this is the difference. It occupies all of the binding sites, so it's completely saturated. The hemoglobin molecule is completely saturated with oxygen. Here, the hemoglobin molecule is completely saturated with oxygen, but the number of oxygen molecules, they decrease from 18 to 16 to 8. Sorry, they decrease from 16 to 8. Okay, here they're completely saturated, so the saturation curve remains the same. But for concentration, again, it decreases from 16 to 8. Okay, the number of molecules decreases from 16 to 8. So there is a decrease in this curve. Okay, you can't see it in the curve, but yeah, you can see it in this curve. Did you guys understand? Um, Okay, so basically, this is the one of the main parts of this lecture. Low affinity for O2, more pressure is needed. Okay, if you don't like something, so you have a low affinity for O2, okay? You don't like something very much. You're being pressurized to take it, okay? More pressure is needed. So what happens? You can see from the curve itself, like, guys don't memorize these curves, because if you memorize these curves, you're gonna eventually mess up in the exam, okay? You can't remember all of this with all the other lectures, okay? So you have partial pressure here. More pressure is needed to occupy the HP. So pressure increases. It moves to the right. Okay. More pressure is needed to occupy. Hemoglobin shifts to the right. Okay. Low affinity. You don't like something very much. Okay. You're being pressurized to take it. So you need more pressure because you have low affinity and you're going to shift eventually to the right. Okay. High affinity is basically you like something so much, like why would somebody pressure you to take it, right? You don't need as much pressure to take it. So less pressure is needed. And this curve shifts to the left because here the pressure decreases. Okay. This is like very basic. Um, okay. Okay, so decreased affinity is release of O2. Okay, you need to remember this. This is one of the main concepts here. If you have hemoglobin and you have this bound to it, okay? If the hemoglobin starts hating the oxygen, it's just gonna decrease affinity, right? It's gonna be like, oh, I don't like this very much anymore. So it's gonna just throw it out. It's gonna be like, get out of my face. Like, okay, leave. It's gonna release O2. So when we need more O2 for tissues, what happens is our affinity decreases, and this results in releasing more usable O2. Because remember what I said, only free molecules can cross barriers, okay? Uh, so basically, when we need more O2, only free molecules can cross barriers. This cannot cross the barrier. Even if you have like 10,000 hemoglobin bound oxygen, you cannot do anything. Your tissue is going to die. You need it to be free. Okay, so you need the hemoglobin to release the O2. Okay, so you need it to reduce the affinity at the tissue site. Okay, at the tissue site, you need it to reduce the affinity. and this will result in releasing more usable O2, okay, because it can cross the barrier and it can go to our tissue and it can eventually help it. Okay, so this is our next one. We're going to study about pH and CO2. Um, okay, so here, let me just, um, okay, there is a formula, not a formula, there's an equation, that's like CO2 plus H2O, this is reversible. What this does is it gives you HCO3 minus and it gives you H plus. Okay, so how you don't really need to learn it, but it's better if you know it. It's like you take this oxygen, you give it here, you get HCO3, and then you take another hydrogen, you give it here, you get H2CO3. Oh no, let me just you get H2CO3, but ignore the two, okay? Why does the line keep coming? Okay, just remember HCO3, okay? HCO3 minus. Okay, if you have more negativity, okay, you have more acidity. Okay, just remember, acid is bad, right? Acid is bad for everyone. So you don't need acid in your body. Acid causes negativity. Okay, so because you're decreasing the pH, decreasing the pH, okay, decreasing the pH makes it more acidic, by the way. A lot of people get this question wrong because you confuse increasing the pH and decreasing the pH. Increasing the pH does not make it acidic, okay? Increasing the pH makes it basic. Decreasing the pH makes it acidic, okay? When you decrease the pH, it's bad, right? Like, nobody wants acid anymore. So you're going to reduce the affinity for oxygen and you're going to shift the right because you need more pressure now, right? When you're going to increase the pH, okay, 7.6, you're going to increase the affinity, okay? So you're going to shift to the left. Even with carbon dioxide, it's the same because carbon dioxide eventually gives you HCO3, and it gives you H plus. H plus is the main one that gives you acidic and basic. Acidic and basic, okay? This is chemistry. I don't know if you guys remember this, but H plus gives you more acid, and because if you increase CO2, this is eventually going to give you more H plus, and because of this, it's going to be more acidic. If it's more acidic, it's going to go to the right because it's lower affinity because everybody hates acid. Nobody wants negativity. I just remember it. Okay. Okay, this is the Bohr effect, and this, okay, again, where's my, we have this equation here, okay, CO2 and this one. There's a thing, this symbol in the between, it means that if you increase this here, if you increase this or this here, it's gonna go and form this, okay, but if you increase these two, it's gonna go and form this, okay? Okay, so what happens is our CO2, it eventually releases H plus, as I said. This H plus, it goes to the heme protein. Our hemoglobin is a protein. It goes to the heme protein, and it's gonna bind to this side, okay? It's different from the oxygen binding side, but okay. It's gonna bind, and it's going to change the shape, okay? When it changes the shape, oxygen cannot really get in. Like, you can see there's no space for oxygen to get in, and because oxygen cannot get in anymore, you need more pressure. You need to push it in, okay? It's not going on its own. You need to push it in, okay? You need more pressure. So this decreases the affinity, okay? Whenever you need more pressure, you have a reduced affinity. So equation is an equilibrium. Increasing anything on one side causes a reaction on the opposite. So if you add more reactants, the reaction will make more products. Remove the product. Even if you remove this, it's going to be like, oh, I have less here, so I should keep making more, okay? It's gonna go on this side again. This protein is a buffer and H plus bind to it chain conformation. Harder for CO2 O2 to bind. I'm sorry, O2 to bind. We increase the pressure. We increase the pressure because there is reduced affinity. Okay, so in the pulmonary capillary is as oh my god. Okay, it's in the lung, right? And in the lung, CO2 is less and O2 is more. So because CO2 is less, this is less. Okay, it does not form this. No H plus, no CO2, no H plus, okay? And in the lung, you need more oxygen to bind because you need to give it to the tissues, right? So you can remember it that way as well. So this increases the affinity of O2 to Hp. In systemic capillaries, that means in the rest of our body, we have tissues that keep using the oxygen and keep turning it into carbon dioxide. So we have more carbon dioxide, which causes this side to increase, and this increases. If H plus increases, it goes here, it changes the conformation of the heme molecule, and you need more pressure to push in the O2. So O2 affinity to Hp decreases, and you need increased pressure. Okay? And we talked about this. pH decreases, affinity of O2 decreases. pH decreases means acidic. Okay? Okay. Effect of temperature. This doesn't really have a big effect on our whole body because our body temperature is almost always 37 degrees Celsius. But during exercise, when you guys exercise, I think you can feel like the pump, the blood it just keeps pumping, right? You need more blood. And so the muscle it becomes warm, and because the muscle is warm, uh, so let's say this is the muscle, the blood vessel that's supplying it, this is going to be warm too, right? If its surrounding is warm, it's going to be warm too. Now, because it's warm and it's so hot, like when in the summer it's so hot, you don't want to do anything, you're so lazy. So the hemoglobin molecule is like, I don't want to be here anymore. I don't want to hold this. I'm so tired. So it just drops the oxygen, like I don't want to hold this anymore. Like, I'm so tired. It's so hot. Like, take it. So it releases the oxygen. And this is exactly what we want because our muscles are going through strenuous exercise and they need more oxygen to function. So because hemoglobin, it releases the oxygen, it lets go of it. This oxygen in the free molecule type is going to go to our muscles and is going to be used. So here we need less affinity of O2 because then more O2 is released to the exercising muscle, okay? This is in our muscles, okay? Not in the lungs. Less affinity of O2 would be bad in the lungs because you need O2 to bind in the lungs. Otherwise, you don't have enough O2 for anything. Okay. Let's move forward. Okay, effect of BPG. BPG is basically, I think we had a question on this in our exam. Our body increases BPG, so it's present inside, and usually happens in high altitudes. So in high altitudes, you have less oxygen, right? And you can't really breathe. If you have less oxygen and your hemoglobin is just there chilling with all of its oxygen as a reservoir, you're gonna be like, give me some, right? Like, what are you doing? Like, I don't have enough oxygen to take in. I have no free molecules. You're just there chilling with all of your molecules. So it's gonna go to your tissues and it's going to reduce the affinity. Okay, so what happens is BPG, when BPG increases, okay, it's even given like medically, it's not only by like the body, I think. When BPG increases, this decreases the O2 affinity, so this gives us more oxygen to be used in the tissues. Okay, why does this happen again? Because you need decreased O2 affinity so the hemoglobin can let go of it, and this can be used in our tissues. That's why increased BPG will lead to decreased O2 affinity, and this will give us more O2 to use in the tissues. Okay. Um. Okay. Okay. So we talked about this. We talked about the H plus concentration and CO2 concentration. I think we can quickly go over it. You have the equation CO2 plus H2O, which gives you bicarbonate, and the other hydrogen goes here, okay? This one goes here, CO3, okay? And H goes here. This H is gonna go here. This is how I remember it. Okay, this basically, and then this is gonna go, it's gonna go to the hemoglobin, and it's gonna change the conformation. When it changes the conformation, oxygen cannot go in. If oxygen cannot go in, you have to push it in. You have to force it in, like get in, okay? It's gonna get in. You have to force it in, and you have to increase the pressure. So because you're increasing the pressure on this, if you see here, if you're increasing the pressure, it's moving to the right, right? It's moving to the right. So this is what happens in the Bohr effect, okay? Don't confuse this with Haldane effect. We're gonna study that later, but Bohr effect and Haldane effect are two different things. And then you have temperature. As I said, temperature is usually normal. You have 37 degrees. But during exercise, which is really common because even in university we walk a lot. So, like, used to it. So if you walk a lot, eventually you need more oxygen. And So if you need more oxygen, you need the hemoglobin reservoir, which is gatekeeping its oxygen to release it so you can use it, right? So you're going to decrease the affinity over there, and again, decrease affinity goes right, okay? Decrease affinity goes right. You're gonna decrease the affinity so you can have more oxygen that can go into your tissues so that your muscles, under strenuous exercise, can use the oxygen. Okay, then you have 2,3-BPG. 2,3-BPG is usually in high altitudes or when you don't have enough oxygen, free oxygen molecules. Again, the hemoglobin is gatekeeping it, and we need it to let it go so we can use it, right? So it's going to increase. 2,3-BPG increases. Like it's getting really mad right now. Like okay, it's increasing. Okay. Uh, it's like when somebody gets really mad, their BP increases, kind of, right? So here BP is increasing. Like, why are you not giving me oxygen? Okay, why are you taking all of it? Okay, so now the hemoglobin is going to reduce its affinity. It's gonna be like, okay, here you go. Okay, it's gonna give the oxygen to the tissues. Okay, this usually happens in high altitudes. Okay, so the doctor mentioned this, but he mentioned it very briefly during the lecture. Okay. So double Bohr effect or double circulation. What happens here is double circulation is basically two different circulations. And this usually happens in pregnant females. I don't know why I mentioned females, but pregnant females. Okay, so you have the fetus circulation and you have a separate mother circulation. Okay? Okay, so what happened? Double Bohr effect. We learned that the Bohr thing is CO2 plus H plus increases, and then these two increase basically. And they go and they change the conformation of the hemoglobin molecule, and then you have to force the O2 in, right? I'm hoping you guys remember this by then. Okay, what happens here is that, okay, let's make this. This is the fetus blood, and this is the mother's blood, okay? Now the fetus, it used up it can't do anything for itself, to be honest. It's trapped in the belly, right? The belly. You have CO2 here. It produced a lot of CO2 and it needs its mom to like get rid of CO2 and give O2 back. Okay. Now this produced CO2 is gonna go to the mother, right? Now it's asking for help, like can you give me oxygen? This CO2 goes to the mother and this increases the concentration of carbon dioxide in the mom. Because now the mother has her own carbon dioxide and she has the fetus's carbon dioxide. So it just increases the carbon dioxide. Okay. Now, as I said, if you have increased carbon dioxide, so okay. If you have increased carbon dioxide, eventually you're gonna have the whole conformation thing, and your oxygen cannot bind. You have low affinity for oxygen. So what this does is it reduces the affinity. I don't know why this line is here, to be honest. Okay. This carbon dioxide increases, and this reduces the affinity for O2. So the mother cannot have her own O2, and because there's a reduced affinity for O2 in her own blood. Why? Because the fetus released his carbon dioxide into the mom's blood, increased the overall carbon dioxide, and now she can use her own oxygen, which is wonderful. Uh, so now this oxygen is gonna go to the fetus side because the fetus needs oxygen to survive, right? So this one is gonna go here, and because it reduced its carbon dioxide, the carbon dioxide was reduced, right? Because all of it went to the mom's side. So because the carbon dioxide reduced, all of the O2 went to the fetus's side, and it has a high affinity because it has no carbon dioxide. It's basically chilling. It has high affinity, it binds to hemoglobin, it circulates everywhere, and okay, it's released. That's what basically happens. Mm. Okay, there's another thing. It's called inactive hemoglobin. It cannot transport O2 very well. It's basically lazy. It's inactive. It cannot transport. It doesn't want to transport O2 very well. Okay. Um. Okay. So CO, carbon monoxide. Okay, I don't know if you guys heard of this. I think you probably have. Like whenever there's a fire, they say cover your face. Like don't let the smoke go in. Why does this happen? Because the smoke over there, it has carbon monoxide, and you don't want the carbon monoxide to go in because carbon monoxide is very evil. Okay. There's two things it does. Okay. There's two things it does. First of all, it reduces the number of oxygen molecules. It takes their place instead, okay? It's very evil. It's narcissistic. It just wants everything to itself. It's like, you can't come here. This is mine now, okay? This is mine now. This will never be yours again, kind of thing. So it reduces the number of oxygen molecules and it takes their place instead, okay? And it also won't. So, for example, if there is an oxygen molecule that kind of went ahead and took the place, it didn't know that everything was taken by CO. It went ahead and it went to its own binding site, which was supposed to be its own binding site. What carbon monoxide does is he's it's gonna be really mad about, like, why did you come here? I told you not to come here, right? No, I won't let you go ever again. That's it. I'm gonna go and hide you in my basement. You're never leaving again. So it increases the affinity. Affinity was, what was affinity? Affinity was basically um the link or bond between the hemoglobin and oxygen, right? So because you increase the affinity so much, it's going to increase it so much, it's not going to let the oxygen leave anymore. Okay? So one thing it does is it reduces the number of oxygen molecules and takes their place. The second thing is, if it happens that the oxygen molecule does take its own place, it never lets it go. It has increased affinity. Okay? In the basement. Okay. We have three containers here. This is the normal one. So you have hemoglobin, you have oxygen, and the oxygen binds to the hemoglobin, 100% saturation because all of the binding sites are filled, okay? Then you have this one, you have 50% anemia. In anemia, you have a low RBC count, so this kind of huffs, like I told you in the two graphs. Like I told you in the two graphs, the saturation curve and the curve I explained to you, how it reduces from 16 to 8. And in the saturation one, it's still like completely saturated, right? So it's 100% saturation. So even in 50% anemia, when the number of hemoglobin molecules are reduced, you can still see that the oxygen comes and binds to all of the binding sites still, right? So there's still 100% saturation, okay? The concentration lowers, but the saturation is still the same. Okay, but now you have carbon monoxide poisoning and it wants everything to itself because I don't know, and it has 50% saturation. Why? Because reduces number of oxygen molecules and takes their place instead. So it took the oxygen place instead. So now you don't have complete saturation, right? Like look at this, it's 50% saturated. It has like two oxygen molecules, two carbon monoxide molecules. Okay, and yeah, that's what it does. Yeah. Okay. So these curves kind of like go ahead, go with these. Okay, so first of all, we're going to look at the normal one. You have normal hemoglobin with 100% saturation, which is our normal curve here. You have a normal number of hemoglobin molecules, you have normal saturation. This is our normal blood, right? It says normal blood. And then you have this part, you have the anemia one. Gonna go see the anemia one. Okay. In the anemia one, uh, here, it basically went down because even though you can see here that it still achieved like a saturation, but it went down. Why did it go down? Because in anemia you have a less number of hemoglobin molecules, so even though there's going to be complete saturation, you can't really bind to that as many as you were able to in the normal one because hemoglobin molecules reduced overall, but it still attains like a saturation. Okay, in carbon monoxide, what happens is you have 50% saturation and you have with O2, and 50% is with O2 and 50% with CO, carbon monoxide, okay? And this is the curve. It shifts to the left. Why does it shift to the left? Is because carbon monoxide itself, it has a 300 times more higher affinity, okay? That's why it's so fast at taking the sides. And even with oxygen, if oxygen binds to its site, it's gonna say you're never leaving again. You're in my basement, right? It increases the affinity for it. So increased affinity is towards the left. Okay, you can still reverse it, by the way. It is a reversible process. We need more PO2. If you increase the pressure of PO2, you can get rid of the carbon monoxide and you can go and take the place instead. You can break into the basement. Amazing. Uh, okay. I don't know if you have to know this. But you don't need to know the text, by the way. Basically, in the normal venous blood, when you take normal blood, when you do like a blood test or you're donating blood, you can see that it's like really, really dark, right? So this is the normal bluish purple blood in your venous blood. But when you have a carbon monoxide poisoning, this is how they detect that you have carbon monoxide poisoning, because your blood, it turns a really, really bright red. It's like a cherry red, okay? You can see here. That's how you detect carbon monoxide poisoning, basically. Okay. This is another inactive HP. Okay, so here two processes are happening. One is oxygenation and one is oxidation. Okay? Don't confuse these two. Oxygenation is adding oxygen, okay? Um, let's see. So for example, oxygenation, it reminds you of generation. So the generations keep on adding, right? Oxygenation is just adding oxygen, okay? So here you have a, one second. Okay. Here you have a hemoglobin molecule and it's deoxy, means there is no oxygen, okay? We're gonna go ahead and we're gonna add O2 to this and we're gonna oxygenate it. And when we oxygenate it, uh, you're gonna get oxyhemoglobin, which means there is a oxygen molecule present in the hemoglobin. Then we're gonna go ahead and we're gonna release the O2. You usually release O2 when you need the oxygen molecules for tissues, okay? So this process is in the lungs when you attach the O2, and then you transport it all the way to the tissues, and then that's where you release the O2. Okay, and then the oxidation happens and you get this. The second part is oxidation. Oxidation reminds you of date, so it changes it to 2 to 3. Kind of like today's the second, it changes it to the third. I had POD on the third. Okay, so this is basically poisoning in a way because it's kind of irreversible. Uh, mm, okay, it's poisoning because it's irreversible. What happens here is that you have the deoxyhemoglobin, right? You have the normal Fe2 plus. Fe2 plus, okay? You're gonna have an oxidizing substance, anything that oxidizes it, and you're gonna change it to Fe3 plus, okay? Fe2 plus to Fe3 plus, okay? And no oxygen can bind here because you changed the iron thing. You changed it, okay, you changed iron from 2 plus to 3 plus, so no oxygen can bind here. If no oxygen can bind here in the first place, it cannot give anything to the tissues. So not kind of, it's very bad for you. And the only way you can reverse this is if you have the methamo methemoglobin reductase enzyme. So this Fe3 plus is basically methemoglobin, and you need methemoglobin reductase. You oxidized it, and you need the reduction, right? Methemoglobin reductase to change it from Fe3 plus back to Fe2 plus. Okay. Uh. Okay. Let's read this. HB hemoglobin with Fe3 plus is irreversible, cannot bind O2 even when PO2 increases. Okay. You can see it's repelling, right? It cannot bind PO2. So we need methemoglobin reductase to reduce Fe3 plus to Fe2 plus. And this is present, there should be an N here, in our blood. Some people, though, they lack this enzyme, which is very bad because they have a difficulty in breathing because O2 transport is eventually affected. You cannot bind O2. So you have a difficulty in breathing and doing oral tasks. Okay. Do you guys understand? Mhm. Okay. So even in carbon dioxide, we're gonna have two forms. Second. So even in carbon dioxide, we're gonna have two forms, okay? Well, not really two forms, two categories. There's three forms. In oxygen, you have two forms. You have physically dissolved and chemically bound. Here, the physically dissolved and chemically bound are like two categories. You have three forms. This is um, this is literally carbon dioxide, physically dissolved. Chemically bound is, you can see these. This does not look very easy looking. I don't know. So this is bicarbonate. This is carbamate. Okay. move forward. Okay, so what happens is, again, this equation will not leave you. Okay, this equation, what happens in this equation is you have bicarbonate, okay, and bicarbonate is the chemically bound, one of the chemically bound ones. This is the physically bound one. And another thing, as I told you, hemoglobin is a protein, right? This is the amino proteins are made of amino acids, and this is the amino acid structure. So amino acids basically have an amino group, which is here, basically. So carbamate, like, okay, carbamino is basically carbamate. It's like carbamino is basically carbamate. Okay, so you have carbamate from this. Carbamino because amino acid has this amino group, and so it's basically carbamate. Okay, and then what do we have? Okay, this is kind of you guys need to draw this and learn. It doesn't take a long time, but anyways, let's go over this. Um, okay, so you have your tissue here, right? Yes. Okay, you have your tissue here, and this is reversible because it's either tissue or lung, but we're gonna take tissue. You have your tissue here. So as I said, in your tissue, you have a lot of carbon dioxide, right? So this carbon dioxide, it's gonna go ahead and your erythrocyte is carrying your oxygen and whatever, but we're just talking about carbon dioxide. So your carbon dioxide is gonna go from the tissue and is gonna go into the erythrocyte. Okay, it's gonna go into the erythrocyte. When it goes into the erythrocyte or RBCs, they have hemoglobin, and hemoglobin is a protein. So what does it give you? It gives you carbamate, okay? So CO2 plus Hb protein, it gives you COO minus. It gives you carbamate, okay? Again, in erythrocytes, in both erythrocytes and plasma, actually, you have H2O, okay? What this H2O, I think you've learned this, honestly, you should have learned this reaction until now. It gives you HCO3 minus. This is bicarbonate, and even here you get HCO3 minus, okay? Oh, okay. Okay. You have HCO3 minus here, and you have HCO3 minus here. Now, the thing is, in the erythrocyte, is that it has special features. It has carboanhydrase. It's an enzyme, and this is going to speed up the process of forming HCO3. So it keeps forming HCO3. So HCO3 increases. Okay. HCO3 just keeps increasing, right? Another thing that happens is that there is H plus, and um, so RBC has again HB, and HB is a buffer, right? It's a protein, so it's a buffer. So it binds to H plus. It's gonna go here. It's gonna bind here, and it's gonna go and remove it. It's literally gonna throw it out. So this part of the reaction is gonna be like, oh no, we have less H plus, and it's gonna be fooled. It's gonna be like, we have less H plus. We need to make more H plus because yeah, because that's what we're doing. If you have less H plus, then we're useless. So it has to make more H plus, okay? Now it's gonna be full, it's gonna keep making more H plus, and along with H plus, it's gonna keep making HCO3, okay? Now you have bicarbonate increases by carboanhydrase, and it increases because of the buffer part, because Hb binds to H plus, and it removes it. So this part of the like I told you, right? CO2 plus H2O gives you HCO3 plus H plus. Okay, sorry. Okay. Like I told you, if this lowers, it's gonna go here. It's gonna go to this side. So it's gonna keep making bicarbonate. Now you have more bicarbonate in the erythrocyte than in the plasma. Again, we're gonna use the rule of diffusion. What happens in diffusion is it goes from a higher level to a lower level, right? So now we have to remove not remove, this has to go from the erythrocyte to the plasma. But this happens with an exchange transport kind of thing. As HCO3 moves out, Cl goes in. Okay, you can see this has negative and this has a negative charge. So in that sense, it maintains a charge balance as well. So HCO3 moves out, Cl goes in. And at some point, it's not regulatory really. So at some point, HCO3 here is going to increase because this is making HCO3, this is releasing HCO3, so this is going to increase, and this is going to reverse the process. The more HCO3 goes in, the more HCO3 goes inside, and with this, Cl minus moves outside. Okay? This is what happens in this slide basically. CO2 can diffuse easily through membranes, binds with hemoglobin, has carbamate, binds with hemoglobin, has carbamine of CO2 or carbamate. So in the RBC and plasma, it's found in bicarbonate. In the erythrocyte, it's faster and quantitatively more. Why? Because of the carbohydrates and because HB is a buffer, it binds with H plus and it gets rid of it. So the whole equation, it keeps moving to HCO3 side. So there is more HCO3 inside the red RBC, and it's going to be exchanged with Cl minus. So it's just gonna go out in exchange for Cl minus in, and the whole thing. And if this increases outside, reverse is gonna happen. This is gonna go back in, and Cl minus is gonna go back out. Okay. Um. Okay. I think. Okay. So as you can tell from this curve, if you compare it to the curve we had for oxygen, it went like this, and it went for the chemically bound. It went like this, right? There was saturation here. Okay, like no more oxygen molecules could bind. Over here, there is literally no saturation. Okay, it does not reach saturation. So, here again, you have the physically dissolved curve. In that one, you had parallel, right? You had like, and then you had like a constant difference between it with increase in pressure. Over here, there's none of that. There is no difference. The difference between both curves isn't constant. The pressure difference is low. Okay. Again, you studied this yesterday. Right to left stent, arterial PO2 is lower than alveolar, and then eventually, basically, carbon dioxide, it does not have such a huge change in pressure. Like, uh, let's say if you just change a little bit, it's not gonna cause a huge change in concentration. Unlike O2, okay? So everything is wrong with O2. Our PCO2 is like the normal sane gas, okay? If you have like a small change in partial pressure, it's not gonna be like overly dramatic and just change all of its concentration, okay? So there is like only 5 mmHg pressure and it does not change much, okay? Because I think, oh my god, lines. So there's a steep curve and it does not change much pressure difference for CO2. In O2 though, there was like a huge difference because it's dramatic. Okay? It's like everybody needs me, so I'm going to be dramatic. Okay? Because what are they gonna do without me anyways? Acidification causes CO2 release from mining site. Okay. What happens here? Again, the equation. I hope this does not haunt you. What happens here is that you have forms of carbon dioxide inside the fluid, right? You have chemically bound forms. So it's bound to hemoglobin. And you want to get rid of it. Like when it comes to the lungs, you want the carbon dioxide to move from the hemoglobin so you can take it out of your body, right? You can, as a form of gas, you can release it. How this happens is basically uh, you're gonna add H plus. This is like an experiment thingy, okay? You're not adding H plus into yourself. Into this, you have HCO3 minus, let's say. Okay? And you're gonna add H plus. This is going to bind to this, and eventually this is gonna give you CO2, right? This is gonna bind to this, it's gonna give you CO2. And this CO2 is gonna come out as a form of gas. Okay? It's also going to release H2O, like when you like breathe out, you can feel like droplets of water, like water vapor ish kind of thing. Okay? That's the H2O, but like you don't need to know that. So CO2 comes out and CO2 decreases. So then the curve, it shifts downwards. I think in the PPT, the old PPT, it said right, but it's down, okay? CO2 is a gas, it leaves the system, it's going to leave the system, and CO2 in the fluid decreases, and the curve shifts downwards, okay? You can see the curve is shifting downwards. It's still starting at the same point. It's not shifting here. It's not shifting here. It's just going down. Okay? And this is so important. This is very, very important because I remember, I think we had this in the exam as well. And yeah, okay. So two-thirds of the CO2 is transported in the form of bicarbonate, okay? Bicarbonate is HCO3 minus. This is bicarbonate. 2/3, like most of the carbon dioxide is transported in the form of bicarbonate. The rest of it is carbamate and physically dissolved. Okay, remember this. If you forget this, I'm going to haunt you. Literally. Okay. Okay, this is one of the last parts of the lecture. Here we're going to talk about the Bohr effect and the Haldane effect. We already talked about the Bohr effect. In the Bohr effect, you had carbon dioxide and H plus increasing. Here you can see you have carbon dioxide increasing, you have H plus increasing. H plus eventually goes and binds to the hemoglobin site, changes the affinity of oxygen because then it changes the whole shape of the hemoglobin protein. Okay, so you need to push oxygen in. Okay, because you push oxygen in. That means you need more pressure, which means you have decreased affinity. And the Haldane effect, it's kind of opposite, right? The Bohr effect, by the way, usually happens in the tissue because in the tissue you have a lot of carbon dioxide. You need to get rid of it. You have a lot of carbon dioxide, and uh, you have a lot of carbon dioxide, and you need oxygen. As I said, you need to reduce the affinity of oxygen with the hemoglobin, so hemoglobin can get leave the oxygen so that you can take it, right? You need a free form of oxygen to use it. So because carbon dioxide is so high in the tissue and it's going and binding to the hemoglobin, it's making it let go of the oxygen, okay? Because, and then you have the oxygen that was let go, the free oxygen. It's too much oxygen, okay. Because you let go of the oxygen, you have more available to your tissue, okay? That's why you have the Bohr effect here. Okay, in the lung, okay, the Haldane effect happens in the lung. Okay, so basically you have instead of high CO2 and H plus in the lung, what do you have high? You have high oxygen, right? So high oxygen binds with H2. It easily binds with H2, right? Because there's so much oxygen. It easily binds with H2. And so the affinity of CO2 and H plus decreases. It's okay. Okay. So this is basically the Haldane effect. This happens in the lung because, okay, let's summarize this, okay? I don't know if it's confusing you guys, but okay. So you have your lungs. Let me draw a really goofy drawing. You have your lungs, okay? In your lungs are responsible for reoxygenating your blood, okay? What would that mean? That would mean it's taking in more oxygen. You have a very, very high amount of oxygen in your lungs, right? And in your, say, in your tissue here, you have very high carbon dioxide because your tissue uses the oxygen and it needs to get rid of the carbon dioxide. And this is the blood flow between them. It has like RBCs with the hemoglobin. Okay, it keeps going here and here and here. So all of the oxygen, because it was so high, all of it bound to the hemoglobin easily. And this decreased the affinity, like it's gonna go back here, carbon dioxide. This decreased the affinity of CO2. Why do we need to decrease the affinity of CO2 here? So that the hemoglobin can leave the CO2 and the CO2 can go into the lungs and then come out, okay? Like we breathe CO2 out. That's why we need to decrease the affinity of CO2. We need to decrease the affinity of CO2 in the lung so that the hemoglobin can let it go so we can take it out. In the tissue, it's going in, right, with all of this oxygen, and here you have a lot of carbon dioxide. This is gonna bind to the this is gonna bind to the hemoglobin, and this is gonna reduce the affinity because you change the conformational change and everything. It's gonna reduce the affinity. Why does this reduce the affinity near the tissue? Because you need the oxygen to be let go. Why? Because you need the tissue to use it. So I'm hoping it's clear. I'm not sure. Okay. Is it clear? Okay. Clear. Okay. Uh, this is literally lung breathes in oxygen. Okay, if you guys have any questions, like let me know. I think this is one of the last parts. No questions? Okay. This was like the very last. Okay, no questions. This was like the very last slide. It's literally just a summary of it. But when I was reading it, I thought that you guys may be confused about the first point, so I'm just talking about the first point here. So in the first point, most of the oxygen transported in the blood is bound to hemoglobin, right? Because first it's bound to hemoglobin, then it's transported, and then it loses the affinity, and then after losing affinity, hemoglobin basically throws it away because it does not like it anymore. Okay, it's throwing the oxygen away, and then the oxygen goes to our tissue and we can use it. Okay, and the maximum amount that can be bound is called the O2 capacity, like we talked about this. Okay, O2 capacity is the maximum amount that can be bound. It's total capacity, right? O2 capacity. Okay. O2 saturation is the amount combined with hemoglobin. Okay, this was the confusing part. I was like, what is this? Is the amount combined with hemoglobin divided by the capacity and is equal to the proportion of the binding sites that are occupied. Okay, all of that. Basically, oxygen saturation, it does not necessarily mean that all of the hemoglobin oxygen binding sites are fully occupied, okay? You're just by saturation, you're just saying that okay, how many sites are occupied? You're telling them. You're like, okay, for example, your total sites or the total capacity that you can have is 10. Okay? That's your total oxygen capacity. Okay? And you used five of them. Okay? You saturated five of them. So your saturation is gonna be 50%. Okay? Because you used only five. So you saturated half of them. Okay? Okay, another example is that if your total sites or O2 capacity is 10, the maximum number of sites you can bind to is 10, okay? And you saturated 10, which will mean that you have 10 on 10, which is 1. You convert this to percentage and you get 50%. Again, you convert this to percentage and you get 100% because your total capacity was 10 and you saturated 10. Okay? That's basically the saturation is the amount combined with hemoglobin. Amount combined with hemoglobin is the bound oxygen that is combined with hemoglobin. You have 5 combined with the hemoglobin divided by the capacity, and your total capacity was 10 and is equal to the proportion. Okay, this is basically 50%. Okay, that's it. Do you have any questions? Let me read the other ones. Oh, okay. Sorry. Okay. So here, the O2 dissociation curve is shifted to the right. That is the O2 affinity of the hemoglobin is reduced. So this was our curve. This was our pressure. Okay. The pressure in all of the curves is on the x-axis, by the way. Only the saturation and the concentration keeps changing. Okay, sometimes it's concentration, sometimes it's saturation. So like, keep that in mind. Uh, anyways, so it shifts to the right. That means you need more pressure. That means there was a lower affinity. Okay. So that is the O2 affinity of the hemoglobin is reduced by increases. Okay, these are the factors we talked about. Increase in PCO2, it causes H plus. Okay, H plus also decreases the pH, by the way, because that makes it more acidic. Okay, increasing pH is basic, decreasing pH is acidic. Don't make that mistake. PCO2 and also H plus goes and binds to carbon. Sorry, 1 second. It goes to the hemoglobin and it changes the shape of the hemoglobin protein, and you cannot fit any more oxygen in it, and you need to force it in, which means you need to increase the pressure again. Okay, it shifts it to the right again. Temperature, so again, as I said, your body is almost always normal, except for when temperature is usually a factor used in exercise and when your muscles under strain, and then you need like a lot of oxygen. So because your muscle is gonna be in under strain, your temperature is gonna increase. Your and the blood vessel there, the RBCs inside, the temperature for them is going to increase as well. So in the summer, when you're like really, I'm sorry if I'm speaking too fast, but if you're temperature increases in the RBCs, it's gonna be really hot. He's gonna feel so hot, like he's so tired, like I don't wanna hold this oxygen anymore, like take it away. Okay? So the affinity decreases again. And most of the CO2 in the blood, BPG is usually in high altitudes, and if BPG increases, it reduces the affinity for O2 in the hemoglobin molecule. Why do we need this? Because high altitudes have lower oxygen that we can use, And we need the reservoirs, hemoglobin reservoirs, that are low-key selfish. We need them to give us O2. So we're gonna increase our BPG so that they can give us O2. Most of the CO2 in the blood is in the form of bicarbonate with smaller amounts as dissolved in carbamino compounds. Okay, this is the thing I told you. Most of it is in the form of bicarbonate. Please remember this. It can probably come up. Okay, most of the CO2 is in the form of bicarbonate. The CO2 dissociation curve is much steeper and more linear than the for O2. Okay, we talked about how it was like this. CO2 one was like this, and it was never a saturation. But in the O2 one, the O2 one was like this, much steeper. The PO2 in the some tissues is less than 5 mmHg, and the purpose of much provide an adequate gradient for diffusion. Okay, if you have higher PO2 here, you have lower, like in your blood vessels. If you just got a blood vessel from the lung, not directly from the lung, if you got it from there, you're gonna have a higher PO2 here, and in tissues you're gonna have a lower PO2, so it can diffuse, right? After it gets comes out of the hemoglobin and turns into a free molecule, because you need free molecules to cross membranes. Okay, factors determining O2 delivery to tissues include the blood O2 concentration and the blood flow. Okay, this is basically just talking about the gradient. And we talked about this. Okay, do you guys want to answer these questions or like, because nobody's answering? Okay, or like, do I just leave it and then we can do it later? You guys want to answer? Okay, okay, you can try. So just a patient was administered BPG. So BPG. He was administered BPG. How does it affect affinity? Okay. By the way, this question was in our exam. I literally just took it from there. So, what does it do? Does it increase affinity and shift it to the right? Does it decrease affinity and shift curve to the left? Does it decrease affinity and shift to the right? Does it increase affinity and shift to the left? Okay, so the first thing when you solve this, just remember that see that decrease affinity and the side is right, okay? Like if you have decreased affinity, where does it go? So you can like directly eliminate it from there. Okay, somebody answer? C. Yeah, it is C. Yeah. So it decreases affinity and it shifts to the right. Like we talked about this, it increase in BP. Yeah, it was C. It was C. If so you guys were listening. Good. Patient was administered BPG. As I said, usually it increases in our body, but we can also administer it. So BPG was increased. Okay? If BPG was increased, you're gonna lower the O2 affinity, which is gonna give us more O2 in the tissues, like I talked about here. Okay? Again, decreased affinity means you need to push the oxygen in because the hemoglobin doesn't like it anymore. Okay? It hates it, low-key. So you need to push it in. That's why decrease affinity, it shifts to the right. If you said decrease affinity and shift, you can just eliminate this one, okay? Because decreased affinity does not go to the left, it goes to the right. And increased affinity, it does not go to the right, it goes to the left. So these two would just go away. These two would be the main two ones. So it was a 50-50. And then if you know the answer, you know the answer. Okay. Most of CO2 is in the blood is in the form of what? Carbon monoxide, bicarbonate, carbamate, physically dissolved, or none of these. Okay, there's one B. Uh-huh. Okay, so it is B. Okay, I stressed over this too much. Guys, remember, most of the CO2 in the blood is in the form of bicarbonate, please. Okay, this one. Okay, so in the hemoglobin O2 dissociation curves, the curve from A to curve B could be caused by, why does it move from here to here? Why does it shift to the right? Why does this line keep coming? Okay, why does it shift to the right? B for this one? Maybe A or C, D or E. A or C or D. Fetal hemoglobin. Okay. Okay, so basically the answer is C. Okay. Uh, this is because it moves to the right. Why does it move? Okay, where did the graph go? Let me just go back to the graph. Okay, in this graph, you can see that the line is moving kind of to the right, right? It has more pressure and it reaches the same saturation and more pressure. Because of this, the affinity is basically low. Okay, low affinity. Where would we want low affinity? We want it in strenuous exercise because if you have low affinity in strenuous exercise, you can release O2 and you can use this O2 in your muscles. Okay, it's my arrow. Yeah. Here you can see our body in the muscles. During strenuous exercise, temperature of muscles increases. We need to decrease affinity of O2 near tissues, so more O2 is available. Let's understand. Okay. Okay, next question. Okay. Which of the best describes the Haldane effect? Haldane and Bohr effect are different things. Okay, I think I marked the wrong one. I think it is C. It's not. It looks like the Bohr effect. Yes. Okay. It is C. Uh, oh no. Okay. A decrease in CO2 binding in hemoglobin in high oxygen environments, which shifts the CO2 binding curve to the left. Shifts it to the left. Shifts it to the left. Okay. A decrease in CO2 binding. Okay, yeah. Because in Haldane effect, you have more oxygen binding, so binding to nonsense CO2 loading in system. That's the Bohr effect, yeah. Hmm. Okay. Okay. Methemoglobin. Okay. Okay. Anyone else? C? Okay. Yeah, it is C. Okay, that's pretty good. Easy. Um, or you guys listen pretty good. So, methemoglobin, like oxidized form. Okay, so it's basically what happens. It's basically what happens. You have Fe2 plus, and because of oxidation, you have Fe3 plus, and then you need methemoglobin reductase to take it back to Fe2 plus. In the Fe3 plus stage, no oxygen can bind, so that's bad. Okay, this one. I think this is the last one here. Hmm. Okay, we have one E. A. Okay. Okay, so anyone else want to answer? Okay, the answer is E. Wait, okay. So this is because here it's asking you and containing carbon monoxide, right? It's increasing the affinity. Like if you go, wait, let me go in closer. I told you that if any carb oxygen does get in and goes like, this was my place, it's gonna take it and throw it in our basement. Like, okay, so it's gonna increase the affinity so much that oxygen cannot come out. It's gonna hold it so tight together, right? So it increases the affinity too. That's why it goes to the left first, and then it's gonna go and it's gonna saturate. Okay, it's gonna occupy all the binding sites, so oxygen cannot really bind to anything anymore. That's why it's like very low too, and like, alright, that's why it's like very low as well, because most of the sites are occupied by carbon monoxide. Carbon monoxide does two things: take O2's place, and it high affinity for O2. High affinity for O2. Basement. Yeah. Okay, so somebody answered this right? Okay, that was it, I think. So please scan the code, and thank you so much for attending today.